Okay, today we're going to talk about Wolbachia, which is my favorite topic. Can you hear the excitement in my voice?
In 1924, there's two guys, Hertig and Wolbach. And around that time, people were going around and looking at insects. They would take an insect and then they would smash it, put it on a microscope slide, add gimsostain. Gimsostain, which just stains bacteria. And then they would look for bacteria inside of insects. Okay? And the reason they cared about this is because around late 1800s, maybe 1890s, I can't remember the exact day, but around late 1800s, people had just figured out that mosquitoes spread filarial worms. So that's like elephantiasis. And they also spread malaria. So this was Ronald Ross who figured that out. He won the Nobel Prize. So a lot of people were discovering that insects spread diseases. And ticks, they would take ticks and they would smash these and they'd find bacteria in the ticks. Okay? And this was interesting because if you were living in places like Montana, you were likely, if you found a tick on you, you were likely to get this disease called Rocky Mountain spotted fever. And that would kill you. Okay? So they wanted to know the source of Rocky Mountain spotted fever. So what they would do is they would go around and collect insects, like I said, smash them and look for bacteria. And then if they found bacteria in the thing, they could say, well, this is maybe what's causing the sickness. Okay. So that was kind of the theme of what's going on in the early 1900s. So they're just taking insects looking for bacteria. So these guys, Hertig and Wolbach. Wolbach is famous for typhus. So typhus is spread by, a bacteria in the body lice. So you have body lice. That's Wolbach made his name in typhus. And he mentored a student named Hertig. Okay, and Hertig took Culex pipiens mosquitoes and he smashed them and he found this bacteria. And he basically wrote this paper that said, well, there's this bacteria. It looks like rickettsia. Rickettsia are bacteria that cause like Rocky Mountain spotted fever, typhus. So there's this bacteria in mosquitoes. It looks like rickettsia, but it's not quite rickettsia, and it doesn't cause disease. So I'm going to name it after my mentor, Wolbachia. So Hertig is the guy who discovered Wolbachia. He found it in a mosquito called Culex pipiens. And he did it by smashing them, looking for bacteria with stains, and then he named it after his mentor, Wolbach. Okay, so that is 1924. Hertig discovers Wolbachia. That's how he did it. So then Wolbachia kind of fades off the map because nobody really cares about it because it doesn't cause disease in humans. Okay, so all these people who were focused on this stuff, they focused their attention to rickettsia because rickettsia causes typhus, and horrible diseases that will kill you. So Wolbachia just fades off the map. And then in the 1960s, okay, there's this guy named Hans Laven. He's German. Okay. He goes around, he's not studying Wolbachia. He's not studying bacteria. He's a insect systematist. He studies evolution and origin of species and things like that. So this guy goes around different parts of Europe. Okay, and collects like 17 different populations of Culex. So he's studying mosquitoes. Okay, so he collects 17 different populations of mosquitoes, and he's kind of looking at like, are these different species or are they the same species? So how would you test if something is a different species? Yeah, you could maybe just like mate them. That's like the simplest thing. If they can mate and produce offspring, oftentimes they're the same species. Okay? So he collects all these different strains of Culex pipiens. 1, 2, 3, 4. These circles are different like colonies. Whatever, up to like 17. Okay? And he does all these complicated like crosses 8 to 3, crosses 5 to 1, crosses 6 to 2. Okay, and he builds this like wheel of what he calls these cytotypes. Okay, and what he discovers is that all these Culex pipiens strains, they look exactly the same. They're morphologically like the same species. But a lot of these things cannot mate with each other, even though they're the same species. So he's like, what the heck's going on? So he names this cytoplasmic incompatibility. Okay, and the reason he calls it that is because he does some microscopy experiments and he figures out that it's nothing in the mosquito that's causing this incompatibility. So it's not something on the mosquito, that wasn't said properly. It's nothing in the mosquito genome that's causing these incompatibilities, okay? He figures out that it's actually something, in theory, he doesn't quite know what it is, but he says there's something in the cytoplasm that's making these cytoplasms of the sperm and the egg incompatible. So that's where the term cytoplasmic incompatibility comes from. It's originated in the fact that Laven figured out that it wasn't something on the mosquito genome, it was something floating around in the cytoplasm. So at that point, I mean, there could have been multiple possibilities. It could have been like a bacteria, could have been like some kind of like a toxin protein thing. It could have been like a virus. Could have been a bunch of things that float around in the cytoplasm. But he wasn't sure what it was, but he coined the term cytoplasmic incompatibility. So that's how Hans Laven discovered CI in 1967. He also, so this is in 1967. Okay. Hans Laven is the first guy to use this for biotechnology. Okay. So what he does is he knows that Culex pipiens is a vector for filarial worms. So that means Culex pipiens mosquitoes transmit elephantiasis, okay, in horrible third world countries and things like that. So he thinks, well, because there's these different strains that are incompatible with each other, what if I took one strain that was incompatible with the strain that's spreading filariasis, and what if I use this strain to do the sterile insect technique? Could I kill off this bad strain in place by using this cytoplasmic incompatibility for the sterile insect technique? Okay, so he does this in Burma, which is now Myanmar. Okay, and what he does is, just like I said, he takes a strain of Culex pipiens that's incompatible with the local Burma Culex pipiens. He doesn't know why it's incompatible. He just knows it might work for a sterile insect technique. Okay, so he sets up a field experiment where he rears a bunch of these males, and he does this experiment and produces this graph. Okay, and this was published in Nature in 1967. A very, very, very famous experiment in Wolbachia in sterile insect technique. Okay, and what that y-axis is, he graphs number of collected mosquitoes. That's on the y-axis. Okay, so 0x. And then on the x-axis, he's got weeks. Okay, so like 1 week, 2 week, 3 week, 4 week, 5 weeks, and he's releasing these mosquitoes every week. Okay, and he produces a graph that looks like this. Right? So what he's showing is that as he releases these mosquitoes in week 1, 2, 3, 4, 5, 6, 7, and then he goes and he collects for mosquitoes in that population, he's seen a dramatic decrease in the population. Population crashes. So he demonstrates, I should also say, he also did there's another part of this graph where he also overlays with number of egg rafts found. So he's also collecting egg rafts. It looks like this. So it matches. So the conclusion here is that, what's the conclusion? The conclusion is that you can use CI, that's the abbreviation for cytoplasmic incompatibility, for the sterile insect technique. It works on mosquitoes. And remember I told you that mosquitoes were difficult. You can't irradiate mosquitoes. So this was a big experiment, because this shows you how to do SIT on mosquitoes, and you can do it using cytoplasmic incompatibility. Okay. So that's why we care about Wolbachia, because it does the cytoplasmic incompatibility. But at that time, in the 1960s, Laven had no idea what was causing CI. He just knew it was there and he knew you could use it. Okay? So then in 1970s, Yen and Barr, he floated around, but I think he finished in California. I think she finished in California too, but I'm not sure if she's active anymore. But Yen and Barr, they were the ones to do the experiments to prove that CI is caused by Wolbachia. Okay, so what they do, what do you think, how could they do that? What do they do? Just guess. What would their experiment be?
Population grows or not.
Right? How would you get a population with and without Wolbachia? If I told you that 100% of culex are infected with Wolbachia, how do you get a population that's minus? You're right. I'm asking you though now, how do you get that? How do you get the minus?
Use a different mosquito that could breed with culex but isn't infected?
No. Super simple question. Don't think so hard.
An antibiotic?
Yes, they use an antibiotic. So, so what they do is you can rear mosquitoes when they're in their larval. Mosquitoes are aquatic, right? And you rear them in these tubs of water, okay? And all you need to do is add tetracycline to the water. And then the little mosquitoes that float around in there will drink the water, and then they get cured of Wolbachia. So they produce a plus and a minus Culex pipiens population, and then all they need to do is they quantify. So this x-axis will be hatch rate. So in CI, the sperm, we'll talk a lot more about this tomorrow, but the sperm combines with an egg, and then these embryos die. So there's a malfunction in the sperm that causes this like sterility, and then the embryos just die. So you can measure CI by measuring hatch rate. That means if a female lays a clutch of 100 eggs, how many of those actually hatch? That's hatch rate. So you get a percent. Okay. If it's 100% hatch rate, there's no CI. If it's a 0% hatch rate, that's 100% CI. There's 100% killing if it's a 0% hatch rate. Okay. And then they do crosses. So one of the crosses is, this is the classical CI, is an infected male to an uninfected female. So this infected male has a modification in his sperm that was unknown at that time. Okay. And they're testing if they knew that CI was related to something in the sperm, right? Because you get sterility and you get these hatch rates. But they're connecting it to Wolbachia. So what they do is they do this cross. That's going to give you hatch rates at basically 0%. Okay. And then they do a cured male, same line, same cross. Only difference is you got antibiotics. You do this cross. And what you find is the hatch rates are like this, up here. Okay? So that proves if it's plus antibiotic, so this is minus antibiotic, this is plus antibiotic. This difference proves that CI is linked to a bacteria that you can kill with an antibiotic. And if you cure the mosquito, the incompatibility goes away. So they are the same species. They're just infected with this bacteria that controls their sterility. Okay? So that is how Yen and Barr linked Wolbachia to CI in the 1970s. Going so fast. Okay. So then in the 1970s, people start to get interested in this because one of the things that you can do is, this is difficult to explain. Imagine, okay, so now here's where I get into this. Imagine you have a population, 50% of the population is infected. I'll do this in red. 50% is infected with Wolbachia, and let's say 50% is not infected with Wolbachia. Okay, so you got a population of mosquitoes. And 50% have Wolbachia, and 50% don't. Okay. Now here's where I tell you the other side of CI. So, if you're an infected male and you cross to an uninfected female, that gives you CI. Okay, because there's a mod on the sperm. Okay. Now the flip side of CI is that if you are an infected female and you mate with that same modified sperm, this female can cure the sterility. Okay, this is called rescue. So CI works like a toxin-antidote system. Okay, what do we know about toxin-antidote systems from where we've studied them before? I've talked about these.
The female has an antidote that gets rid of the toxin.
But what about, where have I specifically talked about these in biotechnology?
Like you mentioned this in Wolbachia.
Besides Wolbachia, outside of Wolbachia, where else have we talked about this? What?
When you were talking about it, when we talked about plasmids.
Yes. Plasmids. And what did I tell you that toxin antidote systems do in plasmids?
It forces it to keep the plasmid.
Yes, it's a selection system. So these plasmids evolve toxin antidote modules because it forces the bacteria to keep the plasmids. So I talked about this and I drew this picture. And I said, imagine a bacteria that's dividing by binary fission. And imagine one of these bacteria gets the plasmid that has the toxin and the antidote, and one doesn't. So this is plus plasmid. This one has no plasmid. Okay? This plasmid is constantly making toxin and antidote. So I'll draw some antidotes and I'll draw some toxins. And usually it makes more antidotes than toxin. Okay? This bacteria that just divided from this, where does it get its cytoplasm from? The mom. So this one is going to inherit some toxins and some antidotes. Okay? But there's no plasmid. So it stops making, as soon as that division goes through cytokinesis and they're two separate cells, it's going to stop making toxin antidote, right? So now what happens is usually it's programmed where the antidote degrades. Now there's no antidote. The toxin now kills the cell, die, and this one's the only one that survives. So a plasmid can make sure that 100% of all daughters inherit the plasmid because it'll kill you if you don't. So the only way to survive is if you inherit the plasmid and you continue to make more toxin than antidote. So this is a selection system, okay, that selects for the plasmid. Now, I just told you that Wolbachia does the same damn thing in the eggs and sperm of insects. So what would you think is going to happen? Like in terms of selection, which one's going to be selected? So basically what I'm trying to say is that
it's going to spread through the population.
Yes, it's going to spread and it's going to be selected for. And the rationale is because which of these females would you rather be? So if you live in a mixed population where some are infected, some are not infected, which of these females would you rather be? Would you rather be one with an antidote or without an antidote? With, because you can mate with Joe over here who's got no Wolbachia and you'll be perfectly fine. And you can also mate with Steve over here, who's got Wolbachia, and you'll also be perfectly fine because you got the antidote. This female, she has to get lucky because if she accidentally mates with a male that has Wolbachia, all her offspring die. Okay? So, if this is one generation, which of these are going to be more likely to survive to the next generation? The red or the green?
Red.
The red. So these ones are all most likely going to survive. And these ones, if the population is 50-50, these ones are going to 50% be completely sterile because they accidentally mated with males. So the next population is going to have 25% green and 75% red. Does that make sense? And that's going to keep going. It's a little bit more complicated because there's other things, but it's going to keep going until it's a very, very, very high rate of infection. Okay? And that's what he meant by the Wolbachia is going to spread. Okay? So you find, that's why in QLEX you find QLEX where it's 100% infected. Okay, now I'm simplifying a little bit. Like there's way more advanced like population genetics and evolutionary biology that I'm not going to talk about. But that's like the simplest idea of CI. So people in the 1970s like mathematically modeled this and told you like, okay, these are the mathematical models. This is going to be like the rate of the spread. It's going to spread. It's going to fixate itself in populations. Okay, so then in the 1990s, this guy named Michael Turelli from California. He's at UC Davis. He's still there. And he was like, okay, look at these models. It's saying it's going to spread. Nobody's measured that. Let's look out and see if we can find this Wolbachia spreading in nature. Okay? So he goes out in, let's see, I'll draw California. Something like that. I don't know where Davis is, something like here. He goes down here and he finds a population of Drosophila simulans that's infected with Wolbachia. So down here in California, there's infected Drosophila simulans. But if you measure up here, there's no Wolbachia in the Drosophila simulans. So the DSIM up here has no Wolbachia, and the DSIM down here does have Wolbachia. So he realizes this is an opportunity for us to like study the spread of Wolbachia in our own backyard. All we need to do is start like doing screening Drosophila simulans for PCR in different parts of California every year, and let's see what happens, right? How would you screen for Wolbachia? How would you do that?
Could you use that same gel that keeps bacteria alive and just attach them?
You could use microscopy. That might be more time consuming.
Would it be easier just to keep a control of uninfected females as you...
What, like cross them?
What?
You mean like cross them?
Well,
how would you know if they are infected?
the ones that you collect?
Yeah, right. So you could do crossing. You could do that. You could do the crossing. That'd be like what Laven did. You could do the crossing. You could do microscopy. There's a quicker way. You're close. Yeah, I mean, like just do a PCR. So like Wolbachia is going to have a chromosome. Just design some primers that match on the chromosome, okay? And then all you need to do is one, extract DNA from the insect. The insect is going to have the bacteria or not living inside of it. So you're also going to get a lot of insect DNA. But you're also going to get some Wolbachia DNA if it's there. And then you just do a PCR on some Wolbachia gene. You got to make sure it doesn't also PCR a Drosophila gene, but that's not hard. That's easy. You just use BLAST, make sure it doesn't match anything else. And then you can just PCR for Wolbachia, okay? So then you would just run a gel. Your positive control would be your infected line. Here's your band. Negative control would be the Drosophila from the north, and then you just do your tests. You collected these at different regions, and maybe you'd see some of these have Wolbachia and some of them don't. Okay? Very, very, very easy. That's it. That's all you got to do. The harder part is going on collecting. Okay, so he did this, did this for, I can't remember how many years, maybe like 3-4 years. And what he noticed, what he documented, is that early on, these places were uninfected. So California looked like this. And then in the later years, started to look like this. Okay? And then in the final years, I was trying to erase the greens. A lot of the greens went away, and basically California was completely infected. Drosophila simulans in California was completely infected with Wolbachia. Okay? So he actually documented and calculated the rate, okay? And it's something like 120 something kilometers per year. Something like that. Actually calculated like not acceleration, but like a speed of the spread of the infection northwards in California. Okay, so why is this useful for biotechnology? That's a question for you guys. What's the significance of that result? That was published in Nature. Nature's the most impactful. It's impact factor 40 in science. That's the most prestigious journal you can publish in. Why is that such a big result?
You can put stuff in Wolbachia to infect things with it.
Yeah, like what if you wanted to change a population of insects to do something you wanted them to do? What if you wanted them to do X? Maybe you could program X into the Wolbachia, and then maybe you could spread the Wolbachia infection. And then all of a sudden now your population of insects all do X, right? Okay, so that's like the importance of this.
Could you do that if the insect population already has Wolbachia?
That's a good question. So can you replace another Wolbachia infection? The answer is yes. But they have to be what's called bidirectionally incompatible. So, and we'll talk about this tomorrow, is not only do you get CI if you have infected crossed to uninfected female, that gives you CI. Remember, because of modified sperm. But you can also get CI if your toxins and antidotes don't match. So let's say you got a red Wolbachia and a rainbow Wolbachia. The TA system here might not match the TA system here. So this is what would be this. If this gives you CI, that's called a bidirectional incompatibility, because you could also do the reciprocal. You could make a rainbow male to a red female, and that would also give you CI. So you have CI going both ways, if that makes sense. They can't mate both ways. That's why it's called bidirectional incompatibility. So if you created a strain that was bidirectionally incompatible, you could spread them into a new population. Okay? But the way that this works is it's kind of like sterile insect technique. You have to release like a whole bunch. Because let's draw out a scenario where, imagine the whole population. Let's say it is 99.99% uninfected. And you release one infected female. Just one. Does she have an advantage having that antidote in this population? Why? There's no sterilized sperm. There's nothing she's going to rescue. She's actually at a disadvantage. So this is the extreme case where what I'm doing is I'm showing you this kind of like a gradient. Like if the population I showed you up here, if the population is 50% infected, like it's easy to imagine why you'd have the advantage. If the population is 0% infected, there's no advantage. So this is a huge question in the Wolbachia field. Let me ask you this question then. If a population of infection, it's got to start from 1 at some point, like, right? So how you ever going to invade a population, right? That's like a mathematical question that people are addressing. So one of the theories is, this gets into the theory. So one of the theories is, or I should also just pre-state that before this, what I'm trying to tell you is that there's like a threshold of infecteds percent in the population. And if you're not past that threshold, there's no advantage to having the CI. If you pass the threshold where the mating probability becomes likely that you're going to accidentally mate with an infected sperm, then it starts to select for itself. But before that point, there's no selective pressure to keep it, okay? So this is a question in the field is, how do you go from here to here? How do you get from 0% infected to the threshold? How do you do that? There's a theory that early on, what's driving Wolbachia into populations might be like a fitness advantage. Maybe there's something else that we don't quite know that the Wolbachia does for the mosquito that helps it. And maybe that is just enough to drive it past the threshold. Then once it gets past the threshold, the CI really kicks in and then it spreads to 100% fixation. Okay, so that's the theory. So we can talk about some of these things that the fitness advantage might be. A lot of people say that like symbionts, one of the roles that's very, very common in symbionts is they provide like nutrients. So you'll hear in cockroaches and termites, they often have symbionts in their gut, okay? And the symbionts in their gut, in the case of termites, provide them with enzyme that can degrade wood. That's how termites can eat wood, is they can't actually just eat it. They have microbes in their gut that can digest it. Cows are the same way. Cows have like these guts, and in their guts, they have these bacteria, and the guts in their bacteria basically ferment and digest the grass that they eat. So that's how like a cow, butter, that's how like a cow will digest food, okay? So that's one possibility, is maybe that the Wolbachia provides some kind of an advantage that we don't quite know about to hit that threshold. Then once it hits that threshold, it takes off with CI. Okay?
Isn't it only inside the mosquito gonads?
Yeah, so you're saying, well, okay, let me, that's a very good point. Let me explain further. Very good question. So you can compare this to the tsetse fly or the termite. They have actual symbionts in their gut that give them like digestive stuff. But if the Wolbachia is just in the testes and ovaries, well, how could that possibly provide nutrients? Well, what happens? That mosquito has to lay eggs. Remember, what's that process called? Vitellogenesis, right? So what if, when you make eggs, what are you essentially making? You're making food for the next generation. You're making food for the embryo. Maybe there's something that those embryos need to survive. Maybe if you have Wolbachia infection in your gonads, maybe Wolbachia are providing some extra vitamin or some extra nutrient that allows maybe more eggs to be produced, or maybe you get more fit offspring, so maybe more survive. Like you could easily imagine how they could be nutritionally supplementing the insect, but not necessarily from the gut, like you're saying. Very good question. Okay. Okay, so Wolbachia spreads. So let me just quick summarize that. There's two big applications, two big apps of Wolbachia, okay? And they're different, they're independent. One is a sterile insect technique. That's different. That's sterilizing males and releasing males. The sterile insect technique, that's one. Second one is what we call gene drive. That's when we're talking about like the population replacement. Okay? So these are two separate applications. In one, you're trying to change the population, and another, you're trying to kill the population. Okay? Two separate things. But they're both regulated and partly controlled by what? By CI, by cytoplasmic incompatibility. Sterility in SIT is based on the CI, and gene drive is based on the CI. The only subtle difference is that in the gene drive, you're trying to release a whole bunch of infected females because they have the antidote. In sterile insect technique, you're trying to release a whole bunch of infected males into a population that is uninfected. Does that make sense? Because this is going to give you CI. And in this case, you're just going to spread Wolbachia. Because Wolbachia, I should also say, I'm going through this so fast. There's so much to talk about. Wolbachia is spread, it's maternally transmitted. So if I told you it was in the gonads, every time that mom lays eggs, every time she lays infected eggs. So when those females and males grow up, they have Wolbachia infections themselves, and those Wolbachia are in their gonads, and then when they lay eggs, it's passed on to their daughters and sons. So Wolbachia is perpetually passed on in the germline. That's called maternal transmission. Okay. Getting good, getting close. Okay. So, two big applications of CI of Wolbachia, because Wolbachia causes CI: sterile insect technique and gene drive, or population replacement. Okay? Now, let me tell you about a unique thing that was lucky that happened with Wolbachia that made it useful. So, in 2000s, and this is one of the papers I signed, people found this Wolbachia strain wMelPop, okay? And they found that Wolbachia wMelPop, so this is from Drosophila melanogaster, and they called it popcorn. And they called it popcorn because it seemed to like pop up in different tissues, and it would actually kill, it was pathogenic to the insect. It would kill the insect. But it would allow the insect to survive long enough to reproduce. So you can imagine if Wolbachia killed the insect, it could never spread because it would be killing its host, okay? But this Wolbachia wMelPop allowed the insect to survive long enough to reproduce, and then it killed it. So why were people excited about that? Well, mosquitoes, to spread diseases, they have to take a first blood meal, and if that blood meal is infected with something nasty like malaria, they will get infected by malaria, and then they lay eggs, okay? So they use up their blood meal, and then they'll take a second blood meal. So the first blood meal, let's say they got from a bird, and let's say that bird had West Nile virus. They'd pick up the West Nile virus, get infected, and then they go take a blood meal from a human. And then what they do is they inject the West Nile virus into that human. Okay, that's how mosquitoes spread diseases. What this means is that, are you ever going to get a disease from a female that's never blood fed before? No, you're never going to get a disease from a fresh female. It's only old females that have had multiple blood meals that spread diseases, okay? So if you could infect a mosquito with the Wolbachia that allowed them to take one blood meal, lay their eggs, and then kill them after, you could eliminate transmission of all mosquito-borne diseases because you could prevent the mosquitoes from taking two blood meals, right? Okay, so that was the first idea. So they took Wolbachia wMelPop, they infected it with mosquitoes, and they showed that it reduced the lifespan. So if this is like a kill curve. So the y-axis is % alive. Okay. And eventually, let's say all mosquitoes, let's say normal mosquitoes die after maybe 2 to 4 weeks. They're all dead. If they're infected with wMelPop, they survive long enough to lay eggs, but then they die. This would be wMelPop. So if you read that paper, you'll see a bunch of graphs like this that are reducing lifespan. So people were really excited about this. And they got a whole bunch of funding from Bill Gates because they said, well, we're going to put this Wolbachia in mosquitoes. It's going to kill them early. It's going to stop the transmission of diseases. And then it failed, and it didn't work. Okay? So that's science. But we got lucky. We got super lucky. There was a guy he's in Portugal. His name is Luis, I pronounce it bad, but Texiera, however you pronounce it. He found out that in Drosophila, fruit flies, if you have Wolbachia, the Wolbachia do something to stimulate the immune system of the Drosophila, of the fruit fly, and it actually reduces their infection rate with RNA viruses. Okay? So the people who were doing this wMelPop project got super lucky, and they saw this research and they were like, well, Wolbachia does something else. It can also like inhibit RNA viruses that are in insects. So they did a pivot, and they're like, well, we're just going to change our strategy. We're just going to say like we'll test if in mosquitoes, if they have Wolbachia, if the mosquito has Wolbachia, is it less likely that it will get infected with Zika or dengue? These are RNA viruses. And the answer was yes. They got super lucky. Wolbachia, if you put it in a mosquito, will basically bolster the immune system of that mosquito, and it will increase the mosquito's ability to fight off RNA virus infections. We still don't understand the mechanism of why, but we know that it works. So now what they're doing is, this is called now transition, their name is the World Mosquito Program. They are doing the gene drive. So what they did is they took Aedes aegypti, which is a very, very nasty mosquito that spreads yellow fever, Zika, dengue, you name it. And they infected it with Wolbachia from Drosophila melanogaster. So that's wMel, Wolbachia from melanogaster. And these mosquitoes seem to not spread Zika, dengue, those viruses. So now what they did is they released these in Australia. And we know that if you release enough of them, if the wMel causes CI, what's going to happen?
It'll take over.
It'll take over. So they've been releasing these in Australia for years now, and the wMel infected Aedes aegypti has taken over. And now they just published a paper in 2018 where there were 0 cases of transmitted dengue in Australia. So then Bill Gates gave them another $100 million, and now they're releasing these mosquitoes in Brazil and in Mexico and in the Philippines and in Vietnam, all over the world, China. Okay, so they're releasing these everywhere now to try to stop the transmission of dengue and Zika. Okay, and all this works in part because of CI. And I study CI. So tomorrow I'll talk about the mechanism of CI.
Their first plan in the 2000s, did it fail because the mosquitoes that had the wMel couldn't pick up dengue?
I don't know. See, here's the thing is with science is if something works, you hear about it. If something fails, you never really know why, and you never see a publication of why it failed. Because it's kind of like people don't publish negative data. It just, all of a sudden you saw them stop talking about this, and you saw them start talking about the virus inhibition. And the virus inhibition turned out to be working. So I think they tried this. I would suspect, my suspicion is that the fitness cost of having a pathogenic Wolbachia is just going to be way too great. Like imagine...
Like they couldn't breed if they're dying.
Right, exactly. Imagine the advantage of if there was a female that did not have the infection or could fight off the infection and could actually take 2 blood meals, she's going to have double the children than one that has it. If she can survive past, does that make sense? So I feel like the fitness cost of having that pathogenic Wolbachia is way too strong, where it's just going to get selected out. It's just never going to work. And I think that was the case.
That makes sense.
So you can't hear it here, but a student makes a comment about how a fit, healthy female is probably going to be much more fecund, have more offspring than a sickly one.
Then just like if you're saying, if you're a big buck, they probably reproduce a lot more than most of the average. Yeah, like 10 times more. Like it's like Genghis Khan. Yeah, it's yeah. I think that's precisely an accurate analogy. Okay, let's end it there, and then we'll go lab.